I need a microphone. I speak very loudly. Where's the mic? Thanks. Welcome to my presentation on Casa Bio, which the linguists amongst you might recognize is the home of biodiversity. And our motto is a face and a place for every species on Earth. Now let me explain. But in order to explain, I have to go back to when I was at school. And on my wall, there were pictures. Pictures of death. Pictures of war. Horrific scenes. I couldn't explain why I had those pictures collaged on my wall. Perhaps I was trying to understand human nature. And in war, there are the greatest gamut of emotions, feelings expressed. Hatred, love, fear. But it's prepared me for what's to come. To an extent, and maybe that's why I was doing it. Genocide is defined as the deliberate and organized destruction, in whole or in part, of a national, an ethnic, a racial, or religious group by a government and its agents. What's that got to do with this place? Anyone recognize where we are? We're in the Kruger, but this is Kirstenbosch. And Kirstenbosch is the seat of the Cape Floristic Kingdom. It's the smallest of the floristic kingdoms, and it occupies that tiny little red spot at the bottom tip of Africa. The area in green, in khaki, is the area covered by the Cape Floristic Kingdom. It's about 600 kilometers wide, west to east, and about 400 kilometers north to south. That's a really small area to have more than 7,000 species of plant. Now, this is what it looks like along the road verges. So even in those little areas left over between fields, between houses, there's biodiversity, and that biodiversity is teeming with life, not just plants, but animals, birds, things subsisting in that area between the road and the fence line. And so I wanted to do a little experiment this morning. I went on Google Maps, and I picked up by the scruff of the neck that little rag doll, and I dropped him at the bottom, Strathspey, the bottom tip of South Africa. What do you think I saw on Google Street View? Not the biodiversity that one might expect from the previous picture. That is a degraded area because it's been mown. I then thought, let's try a bit further north. Well, that's degraded as well. That's grass. It shouldn't really be grass there. Okay, but let's try a different road, a little offshoot from that. Well, this is cleared on the left, and on the right is that same grass that comes up after degradation. Okay, but that's the south coast. And traveling around there 20 years ago, it was magnificent on the side of the roads. I would stop and enjoy the plants and the flowers that were there. But now, that entire area has been systematically degraded for no apparent reason on the road verges. But let's jump across the mountains into the area of Robertson, and we get the same thing. Okay, but let's jump across mountains further. Let's go to the Klein Karoo. Here we are near the Warmwaterberg, and Ronnie's sex shop showed up. It's like looking for Where's Wally, but in this case it's Where's Willie. See if you can spot it. And at the Warmwaterberg, there was the same degradation. And in fact, turning towards the Warmwaterberg, this is a 2023 picture in April, and they had recently not just mown it, but graded it. They had graded the roadside as far as the eye could see. This, I would contend, falls under the definitions of genocide, an organized destruction in whole or in part of a national, ethnic, or racial group. In this case, it's not just one race, it's thousands of races, thousands of species degraded. How does that touch you? Not that much, right? I'm guessing. Well, maybe that's because Stalin said, if only one man dies of hunger, that is a tragedy. If millions die, that's only statistics. So what we need to do is slow down. We need to connect. Perhaps meditate. We need to cogitate on nature, what it really means, what it feels like. We need to bring back the magic of the environment. We need to get our hands in contact with the earth again, our feet, which is why I, in part, walk around barefoot. We need to create ceremony and get in touch with our spiritual selves again. And particularly as scientists, we're divorced from nature. We've held it to high esteem that we are beyond human nature, that we are beyond that which is fickle, that which is to do with the heart. We're head people. And I know very well, having been 20 years at UCT. We need to connect in with each other, we need to connect in with nature, and we need to become intimate again. I'm sorry about that. But what do all those pictures have in common? They're all AI generated. Those are not people there, those are not women. So I think it's appropriate to put that picture there. So let's go back to the building blocks of biodiversity. The building blocks of biodiversity are herbaria. They're museums. And a herbarium is a museum for plants. In it, there are stacks of specimens. And as an example, Kew Gardens has 7.5 million specimens. They've collected that over more than 150 years, which is 50,000 species per annum. One of the people that contributed to the herbariums was a man called Adamson. Not Adamson, Michel Adamson. And he did an amazing work. This is before there were computers. And he did 27 large manuscripts, 150 volumes with 40,000 species in it, a dictionary containing 200,000 words, and 40,000 figures. That's artworks, and 30,000 representing 30,000 specimens. What an incredible man to be doing such an amount of work. And in a time when people didn't live very long either. But all of this has been surpassed to an extent. Because in the early years of something called citizen science, in which there was a company called iSpot, based out in the UK, and they had a terrible interface. It was atrocious. You would take one picture, click upload, and then you would go back, scroll down to where you were, select the next picture, and select upload as well. I think the data scientists, the computer scientists around here would be appalled at how they thought that it was acceptable to delay people, to put people through such rigmarole and not take a few extra hours to make it more efficient. But as a result, we got 166,000 observations per year with that system. Amazing. But iNaturalist came about starting in 2008, and After a while, they got to 50 million records. That was 4.1 million records per annum. And they're now on 162 million records. That's now 50 million per annum. So it started off at a slower rate, but from 2020 on, it's now 50 million per annum. That's extraordinary. It's fantastic. But the question is it enough? The human population is growing at an exponential rate. Is that correct? Scientists amongst you, is it growing exponentially? No, thank you. My apologies on that. It's growing at a very rapid rate. And yes, there's contention that it's going to drop. But there's a relationship between population and consumption. The question is the footprint, is it linear? Well, it's not, because as the population of China increases, the ecological reserve decreases. And part of that is because cell phones increase at a radical rate. It's not just that each person has one cell phone, but each person has many cell phones in their lifetime. In fact, 1.5 billion in 2022. This is creating a drain on the resources like a monster, the cell phone monster. And rawr, he's devouring the vegetation. But not only are our cell phones something tangible like that devouring the vegetation, but what about our putty tats and our hounds? There's 117.5 million kilograms of meat exported from Germany alone that is going to the pet food industry. And this is leading to a man-made catastrophe around the world. Fish, reptiles, birds, mammals, amphibia, they're all showing radical extinction. What can we do about that? We need an exponential solution. So let's look at the impediments to this solution. The first one is funding for observations. Well, if we look at the Large Hadron Collider, something many of you may be familiar with, the cost was $4.6 billion. The ongoing cost is $5.5 billion per annum. Alone, electricity, 23 million per annum. The Higgs boson lifespan, one of the holy grails of using this, lasts less than a trillionth of a billionth of a second. I think that's almost the definition of ephemeral. What about the James Webb Space Telescope? Amazing, allows us to peer into the heavens. The cost, $10 billion. The closest planet is Venus. That's 41 million kilometers away. Our nearest solar system, Alpha Centauri, is 4.1 light years away. In 30,000 years, we'll be 3 light years away, or a heck of a lot of kilometers. Our nearest galaxy is Canis Major, 25,000 light years away. Let's translate that into kilometers. It's 236 and 500. I'm sorry, listen carefully. So, how much has Earth observations received? iNaturalist has received $10 million in 2023. That's less than what the Hadron Collider expends on electricity. The Bioscape project, one of our latest and greatest projects that we're going to put over the Cape, has received $2.5 million in funding. This is probably the single biggest biological observation platform that's coming into existence. This is nothing. This is nothing compared to what the hard sciences, physics received. There's a huge asymmetry. And yet, those stars cannot be touched. I bet none of you have smelt one of those stars. And indeed, they're going to be burning away long after we're gone. But the plants that are here today could be gone any moment now and are disappearing at an alarming rate. So there's a representation of those amounts, except the AI couldn't quite conceive of the differences, so it put them as 3 slightly different sized frogs, 12.5 billion, or billion as it said. Impediment 2 is we don't know what species look like, many species look like, or have incorrectly identified them. So who knows what tree that is? Okay, what's the common name for this tree? Yes, the baobab. Thank you. You're putting everyone out of their misery here. So I want to ask you a question. What kind of leaf do you think the baobab has? Okay, put up your hands. Who thinks the baobab has a leaf like this? No one. Okay, well done. You're correct. Who thinks it has a leaf like this? I'm only giving 3 options, so we've got one person there. So the rest of you all think it has a leaf like this. Well done. Through your abstinence, you've got it wrong, because the correct answer wasn't there at all. It's here. But before I show you that, I want to explain something. So you might have got a clue from the name, Adansonia digitata. And that's what's termed etymology. And etymology is the root of words. And in the case of science, it's the root of biological names, scientific names. So, did you recognize Adansonia? The name Adansonia, where? In the beginning, I told you Adanson was the guy who submitted so many species to the herbarium. But what about digitata? What does that mean? Yes, you have a question about digitata, an answer. Very good, you obviously saw. It's from the Latin digitatus. So, out of these, let's go through these plants again. Which is it? Is it this one, or the one that looks like a hand? That's the leaf of Adansonia digitata, the baobab. So, this is part of Casabio's work to make scientific names more tangible, so that you can remember them more easily. Adansonia digitata, named after that guy Adanson, and digitata, like a hand, leaves like a hand. And that falls under our gamut of education. But how do we see what these plants look like? Well, one of the things we want to do is to digitize the herbaria of Africa. Starting in the Cape, we would use a camera like this, the OMD EM1, which I have right here. Oh, I fell in love 5 days ago, because for 5 years, I haven't had a decent camera that I love. And I dig this camera. It's fantastic. So, it's beautiful taking photos with it, so I'm in love with botanical photography again. And this camera has an option where you can take a high-resolution picture, it shifts the sensor around, stitches it together, and does 80 megapixel images. And then we can zoom in on those plants and see the details, like the little hairs on the leaves. And then we can use those as a representative to identify other pictures that we get. So that's the idea behind that. But impediment 3 is losing habitats rapidly. That's road and rail reserves, agriculture, mining, invasive species, climate change, and poaching. Now, you've heard about the poaching of rhinos, right? How many of you know about the poaching of plants? Can you raise your hands who's heard about that issue? Okay, so there's 2 hands and 4 nods. That'll do. So, Let's go back to road reserves for a moment. In South Africa, we're endowed with one of the largest road networks. There's 750,000 kilometers of roads, 10 meters on each side of the road, gives 15,000 square kilometers. If one were to consider that as a reserve for conserving plants, how big would that be? How big is 15,000 square kilometers? I'll let you know. We're in one of the only reserves in South Africa that's bigger than it. The Kruger is 20,000 kilometers. So we have an opportunity by conserving these road verges, rather than destroying them like I spent the last 20 slides showing, of creating a habitat for plants and animals to not only live, not only thrive, but to move around as climate change causes the temperatures to move. So those plants and animals need to track the coolest space. And those corridors could allow them to move between landscapes. So it's critical. In Europe, they found that 40% of a country's species are to be found in the road reserves. In South Africa, we've got 22,000 species. That should make about 9,000 species. Yet Greg Nicholson walked from Namibia to Cape Town, and he found 640 species and just 700 kilometers of road, of which 63 were on the threatened list. The implications of this is there's going to be far more species in those roads than we expected. And that's what Casabaya's Project Saturn is about. So what we want to do is divvy the road verges up into sections, segments. And at each segment, we put a QR code with a pole. And anyone, the public, can come in, they can photograph that, and then walk a transect, photographing the plants, the animals they encounter in that transect. And then they upload it to Casabaya, and we then know what species there are in that road section. And not only that, we can compare the species from area to area. We'll also know what those species look like, which can inform machine learning, which can have various uses. What about permits? Permits take years and years to acquire. They only allow specific taxa. So this means that we can't understand we can't collect the plants that we want, that we need to do for scientific observations, because we don't have the permit for them, because they want to know beforehand what we're going to find. So this is a problem. So Kermit is the quick permit system connecting conservationists, scientists, and farmers. To illustrate that another way, there's the patch of land. Up top we have the farmers, and on the bottom, the finely dressed scientists. So we need to link all those people up in order to conserve the areas we have left. And let's get now to poaching. Poaching is mainly for succulents. And we need to do some kind of triage. We're at such a critical place. The number of plant seeds per year, you might guess, are a few thousand. Well, they were at one point. In 2018, just over 1,000. But now, in 2022, we're looking at 242,000 species. That's not taken out of the field. That's those that have been confiscated. A drop in the ocean of what's actually being taken. That is scary as heck for people who care, like me, care about each little plant that I encounter. And it breaks me when I see that taken out of the field, when I see that stripped. Here's an example of those plants being poached. And this is what it looks like. When the field was there, the quartz patch was there. On the right-hand side, all those little patches of bare ground, that's where the plants were. Last 2-3 weeks ago, there was a bust. There was a house, and 4 men were found to be handling the plants that had been taken. So this is fresh. Very few people have seen these pictures, and they are shocking. This plant on the left is Aloe hemanthifolia, And one of the culprits that I believe is the issue is that iNaturalist, one of the organizations that's most responsible for how we know what we know about plants and animals and what they look like as a citizen science platform, is iNaturalist, and they are giving away the precise locations. Look at that. Is it surprising that the result was that, when people know exactly where to go to get them? But they've also come up with a system of obfuscating those localities. And here it is. In the red, we have the obfuscated localities. They are randomly placed within a grid. But I guess it doesn't take a rocket scientist, which many of you probably are, to figure out more or less where those plants occur. And if you look at the habitats they occur in, you would be right in assuming that they occur around cliffs and in the rivers, and from that you can pretty readily figure out where they're likely to be. So, CasaBio obfuscates the localities to a 25 by 25 meter grid, and it doesn't show them unless you have the credentials to log in. That's the kind of protectionism we need going forward to protect our flora. We need African solutions for African assets. But we're also losing species at such a rapid rate that people can't be held responsible for documenting sufficiently. We need to use a new technology, something that can go 24/7, something that we can distribute, something that can be rigorous. What are we talking about? We're talking machines, we're talking robots, right? Something like that. Imagine a team of robots going through the field, documenting all the plants they encounter, using machine learning to uncover what that is, to determine what each species is, and then sending that back to the server and bolstering the identity of each species as it goes, like getting better and better at it. That's what we have through the BotBot. That's the potential we have through the BotBot. What about invasive species? The linear routes, the roads, have got Google Maps, as I showed in the beginning. And along those roads, there are invasive grasses. Through machine learning, we can very readily tell where those grasses are and where they're expanding to with time. So we can look at invasions and see and project where they're occurring and where they're going to occur in the future. So this depicts a train doing something similar. Because if we put cameras onto a train together with the GPS, we can not only get a view from the train for a particular location, but because we take a photo 5-10 meters further on and take another one, we can triangulate where the plants are in space. So you not only know where the plants immediately next to the camera are, but you can determine where the plants are, where each position is in space. So we can get a view, maybe 2 kilometers from the train, of where the plants are, and we can, using machine learning, we can determine what species they are, and then do that for entire transects, not just at one time, but through time. As it goes every week, every month, every year, we can look at how not only the plants, but the vegetation changes through time. And that taps into climate change and understanding the impacts of climate change. And so we have to deal with photographs like this, repeat photographs of the same kind of thing. And tying it in with weather stations, which is depicted here, we can start to really get into the drivers of climate change. But in order to deal with this number of photos, we need a critical innovation. That's JPEG XL. Who here has heard of JPEG XL? So JPEG is the Joint Photographic standard on photographs. And JPEG XL is the latest and greatest. JPEG's over 20 years old now. We need something better. So it's high fidelity. It's got both lossy and lossless. And it has so many incredible abilities. But one of the things is that with a series of pictures, you can encode the first one and then just encode the differences between them. So for something like I was showing you here, we don't need to encode each picture fully. Only some of them. There's a whole lot more benefits, but we've implemented this in the latest version of CasaBio because we need a crazy solution for an insane challenge. And 17 years ago, I presented CasaBio's solution to documenting flora, and this is what it looked like 17 years ago, and it's pretty much unchanged. This is the kind of thing that we're doing with iNaturalist, and today that's called citizen science. And our app that we're developing looks something like this, and we've incorporated over 10,000 books with photos from, over 10,000 species, the pictures from books of more than 10,000 species. And machine learning is surpassing intellectual frontiers at an increasing rate. So thanks to the CSIR, we are, through Dorisa, storing our pictures, and who knows how we're going to use the CHPC in the future for machine learning and implementing that. And they're storage for the long term. So I'm really grateful to the CSIR and what they're doing for biodiversity of Africa. And it wasn't lost upon me that one of the aspects of Nikki is they're the high-speed cluster dedicated to research, education, and innovation. And CasaBio's 3 pillars are conservation, education, and research. So there's certainly alignment there. But I want to leave you with one final thought. And that thought is, are you obsolete yet? And I left one group with that without following up with the critical slide, which is this. CasaBio, your friendly biodiversity overlord. Thank you, everyone.
Thank you. Thank you, Dr. Evans. Questions? I'm going to take at least 2 questions, and then we can maybe take it because we're running out of time for lunch.
Brilliant.
And let me take 2 questions for Dr. Evans.
Yes.
I think there was one before that side.
Okay. Yes.
Hi, I have one. So
And then
you are
later.
using pictures of leaves for botanical characterization, but you are a botanist, and isn't it typical to use the flowers only?
So there have been various paradigms through time of how plants are related to each other. And some of the earliest ones were using flowers only. But now we like to use the entire gamut of information that is at its disposal. So what the leaves look like, what the flowers look like, but we're incorporating genetics as well. And nobody's yet, like iNaturalist hasn't incorporated genetics in. So there's huge opportunities to draw in all these different aspects and making it comprehensive. And what about the knowledge of the plants? Where is that put? What about the knowledge of the uses of the plants? Where does that go? So that's a repository that CasaBio is going to take on as well. Did I answer your question? We're using all of it.
Okay, kind of.
Anything morphological, yes?
So, but when you take the pictures,
Yeah.
you're just going to look at the shape of the leaves. They don't define the species because many species have similar leaves.
Right. So one looks at the full thing. You look at, so when I take a photograph of a plant, I photograph the whole plant, I then photograph the flowers, I photograph the leaves, And then when I'm back at the laboratory, I sometimes even use the microscope to differentiate it more critically. So it depends on the taxon. Some things one can, just a photo of the flower or just a photo of the leaves is enough to identify it. But with other things, you need to be very critical when identifying it.
So how would the train model work when you are at high speed?
Oh, thank you, yes. So the answer is that there'll be a confidence rating of the identification. Some things will be well identified. You then use it's called truthing, to go and determine what that is. You can send specimens back if you need to. And then, so near the train, as an example, you might have a high confidence in what that species is. But in the distance, less so, and only certain very characteristic plants you'll be able to recognize. But the critical thing is that because you'll be storing the pictures and their locations through time, you can go at any time, and as you get greater knowledge of that species, you can update the identification. As the model increases in its ability to identify it, you can rerun the machine learning model time and time again, improve it with time. And what's cool about the train thing is that you're getting multiple perspectives of the same plant, and you're getting perspectives through time as well, as it flowers, as the leaves wither. And potentially you can also utilize different frequencies, not just different wavelengths, not just visible light, but you can use infrared and ultraviolet as well, which can be very useful in identification.
Okay, I'll take, are you answered? Okay, I'll take the last question, please.
Hi, thank you for your presentation. I have worked at Sandy before Kirsten Bosch, and I'm quite familiar with most of the things that you have presented, and particularly iNaturalist. I worked a bit on it before. Now my question to you is, I see you are enhancing some aspects of iNaturalist in your Casabayo, but is it not better to then collaborate rather than starting an own individual similar thing on the side? Because literally what you're presenting is what iNaturalist is doing. And again, iNaturalist is run by organization, and they've got a lot of historical background. There are scientists working on it. They've got a huge database from Karoo Biogaps. Instead of doing a replica and starting from the basis, can you not collaborate where you see the gaps that iNaturalists have? Because I think at the end of the day, the aim is to enhance science, make it better through collaboration, whether it's multidisciplinary or transdisciplinary.
Thank you for that. It's a very common question to ask. So bearing in mind that Casabayo has been going for 16 years, quite a few years longer than iNaturalist. And, but besides that, I find it very difficult when, for more than 10 years, I've been telling them that they need to obfuscate localities. And a large part of why we have the poaching issue that we have today is because they're not receptive to changing it. I spoke to the director at a meeting in Reunion Island, and I spoke to her and made it clear that poaching was a huge threat and she needs to obfuscate the localities. It was only years later that that was dealt with. So I come back to that we need African solutions for African flora. That's not to say that there's not scope for collaboration. Of course there is. There's fantastic scope for it. And they have an API. And we have an API. So we can have an exchange. But I'm not going to make the localities of my plants available to the level that they're wanting in terms of geographic precision. Also, they're doing just one small aspect of what we need to do. They're only doing uploading of pictures from citizens. They're only doing the georeferencing of those and some of the taxonomic aspects. But they don't have the capacity at this point to deal with the roadmap of getting exponentially more pictures. So JPEG XL, it's not easy for them to implement. Also, and JPEG XL also allows those wavelengths to be incorporated, multimodal, multi-band wavelengths to be incorporated. So there's huge things that we can do that iNaturalist isn't planning to do. It's got its hands full with what they're doing already. So they have set the trend, they've done fantastically, but we also need to break away at some point, and that's what we're doing. Thank you.
Thank you, Dr. Evans.
Thank you so much.
Another round of applause, please.
Yeah, so cloud native potential. So let's take a journey down memory lane a little bit in terms of infrastructure deployment. But first, let's have a look at the project that we are. What are you looking for? You want the clicker? I want the clicker, yes. We have to insert a USB thing. We can take this USB. There's a USB slot here. Where's the? Just take it at the bottom. Okay, I know these things. Good. Okay, so this should work. Yeah. So this is very much a collaborative project with Eugene over there, who for some reason is wearing a red hat, which is red, but I don't know why. Before Eugene was doing things like sticking post-it notes to his head or whatever the hell he's doing there, he was working with us at the South African National Bioinformatics Institute, which is where I, the person on the right-hand side, have been working for a few decades. And my background is as a research computational infrastructure builder, the regional system administrator at SANBI, and somebody who has then become a bioinformatician, somewhat by osmosis. So I always say that I last did biology in grade 9, but I kind of picked up things along the way from talking to people. And what did we end up doing? Okay, so Eugene had by then moved on to CHBC's Advanced Computer Engineering, ACE Lab. And we got tasked by our collaborators, the Africa Centers for Disease Control, to build a thing called the African Pathogen Archive that more recently has been renamed as AGARI, or AGARI. This is a mechanism to share genomic data within Africa, okay? So if we look at like infectious disease outbreaks, like the Central African cholera outbreaks that are happening at the moment, these things are not confined within country borders. We saw this very dramatically in the case of the COVID-19 pandemic. And there's a need to share data. At the moment, the options are very limited. One can publish data on some data systems, but you know, we saw the consequences of publishing data about the Omicron variant of SARS-CoV-2 in South Africa with incredible economic costs to us because of short-sighted northern policies. And so we need to build a better mechanism. All of the national public health labs are coming to us and saying that we need a way to share things in a way that's much better controlled than just putting things in a Dropbox or emailing to each other, okay? And the vision is that ultimately there'll be hundreds of thousands of data objects representing genomes of different pathogens or parts of genomes, Plasmodium falciparum for malaria, Vibrio cholerae for cholera, SARS-CoV-2, and other things. That it will be multiple sites and geographically distributed so that we can actually support people's demands in terms of retaining data sovereignty. So for instance, if you're in a country that does not want to store your genomic data, that you do not even trust the African Union, but then we can give you that option. Oh, I don't know what that is doing. We can give you that option while... what? Okay, it's right there, thank you. Yeah, so we can give you that option while still facilitating sharing, okay? And then the idea is that eventually different components run in different places. Not everything has to run everywhere. Our starting point was a design that was used in Canada. So a bit of background here, the Ontario Institute for Cancer Research has been building systems for cancer data sharing for some time. And they were tasked by the Canadian government to build a system for sharing their SARS-CoV-2 virus data that became known as VirusSeq. And as we can see here, it was built out of many components. So this is what we call a micro-component architecture. The advantage of building things this way is that different components can run in different places. You can have different numbers of things running. So for instance, Portal speaks to the Arranger query service. If your query volume increases, you can stand up multiple copies of Arranger, perhaps even just geographically distributed or distributed across different computational nodes, and that way you can deal with increasing data, query pressure and so on, okay? So we thought this was quite a nice design, and so we used this as a starting point for our development, and then expansion on top of this. But before we get into that, we have to look at like, how do we build systems that we are deploying, okay? So, traditionally in high-performance computing, in academia, especially here on the continent, we have what often starts off as a single powerful server or a small cluster run by a department. Perhaps there will be, in a small number of cases, the university will realize a need for this and then have a central cluster. Or we'll have, as we've had in South Africa for some time, the Center for High Performance Computing with a large countrywide cluster, right? And then, if we contrast that thing to cloud, then the traditional virtualization environment, even when we do have virtualization available, if it's managed by central IT, then we basically open a help desk ticket, please create a VM for me, then they basically decide whether it fits within their change management window, and eventually you get a VM, oh now I want to change some configuration about it, I have to wait some more, and you end up looking like the fellows here on the right-hand side, okay? So there's virtualization of hardware, and so about a decade and a half ago, each project would buy its own server, and servers would have names like Baboon Serve or Plant Serve, or depending on the project that bought them. And we moved from that to virtualizing things, but treating things still as like long-running servers where the configuration, I remember how I configured it last year, or maybe I don't, okay? The traditional use of virtualization with long-running virtual machines is not actually making use of cloud tooling, the possibilities that it has on offer, okay? Then, if we look at what industry is doing, some people have embraced cloud extensively, and they're using all of the possibilities that one of the cloud vendors offers them. And this means using the different APIs, for instance, Elastic Container Service, Lambda, and all of those things. If you're on Amazon, maybe you're going to be using Google Pipelines and Google Cloud, etc. Okay, and this model, it's vendor locked, so they make it easy for you to deploy, your Lambda functions. But now, if you want to go move over to Azure, you're going to have to rethink how you did your system. And I mean, for these people, which are who are processing maybe commercial data where you know that there's a certain amount of revenue stream coming in, and then if your sales volume increases by 20%, you can justify increasing your compute on cloud by 20%, then to have on-premises hardware that is a fixed size, well, they don't like that, okay? Then the other kind of industry model at the moment is, well, invest in a massive data center, right? And that starts looking like what I just described about waiting for central IT to do things for you. So centralized quite delayed in terms of, and quite inflexible. And then there's been all this talk about hybrid. Run some things ourselves, run some things in the cloud. As academics are embracing the cloud, they're getting close to this model, okay? But it comes with a lot of complexity. And here we have if you remember when they tried to dig that massive ship out of the Suez Canal, So how are we going to deal with that complexity? And we're going to do that by looking at cloud enabling the way that we work. And when I say we, I'm largely speaking to the people who deploy systems. So in other words, not the people who necessarily are running an analysis, but the people who are building the system that will run the analysis. So when we started building, we used OpenStack, which is the same cloud system that's underlying Saboa cloud and the Ilifu cloud that is run by several universities in the Cape region. And where this is, so when I used to do virtualization back in the day with VMware and things like that, I'd kind of go to some environment or using libvirt, and I'd say create a virtual machine, maybe using a dashboard or some command line tools. One of the really nice things about OpenStack and all of the other modern clouds is that they provide an API where you can just say, I want a network, a virtual network, I want a cloud, I want a router, and things like that, okay? And this is basically giving you the same way that people work with the big guys like AWS and Azure, but in your data center, okay? And you don't end up not being able to use a credit card because you forgot to switch your VM off. Real world example. So, obviously with caveats because, we can't all scale to the size of an Azure. And then when we're dealing with this thing, as deployment people, the rule must be that your infrastructure is software defined. So in other words, you really don't want to be in a situation where I've configured the server and I may never switch it off again because I can't remember how I set it up. So all of the setup is defined in code. So the underlying code is a thing called Terraform, and the secret store vault from a company called HashiCorp. It's, and I mean, there are other systems, Vagrant and Packer and so on, but basically this says, okay, you write a description. I need this number of computers. I need them configured in this way. So that gives you a basic underlying layer. Okay, this is your equivalent of buying servers. Then Terraform means that we end up getting a certain pack of things. If we need to change the system that we're running on, we just need to change the Terraform layer because there's a Terraform for Amazon, there's a Terraform for OpenStack and things like that. Everything else will stay the same. We're just going to change that underlying layer. Then comes the big kicker. So we're using a system called Kubernetes. So on top of your virtual machines, remember I said I don't want a long-running virtual machine that sticks around almost like a physical server does. I want to make things deployed as I need them. So we use container technologies to deploy applications, but then how do your containers all relate to each other? What is the life cycle of the container? Should I have more containers as the demand increases? So we use a system called Kubernetes to manage that, okay? Kubernetes is somewhat known for its complexity, but its complexity is there because it's handling the complex task. And I've got an example here of an infrastructure. And these are what you'll have in the Kubernetes cluster, which is, remember, running on some underlying system. You'll have an ingress layer, which basically handles all the incoming network traffic, and then it'll route it based on generally your DNS name. So maybe service 1 will go onto this service layer, service 2 will go onto that service layer, okay? So this is like a reverse proxy that you would have in your network environment. And then service 2 has each of the services has a thing called a deploy, and deploy just makes sure to keep track of the life cycle of your containers. What must be alive, how many copies, etc., okay? And then the actual software is running in things called pods, one or more pods for the application, which is just a wrapper around a container. And then over here, this is just doing some work, and when that all goes away, there's no permanent data. Over here, we have an example where you've got something maybe talking to a database, and then you have a thing called a persistent volume. And a persistent volume is just an abstraction over, this is a piece of storage that I'm going to want to keep around, you know. So all of these things are being handled via a consistent API. If you use the kube control command line tool, which is an interface to Kubernetes API, then you can say get and the object, or, you know, and delete and the object and things like that, and the verbs are the same for everything. In fact, the verbs are the same for nodes as they are for ingress controllers and all of the other things. So you've got a very easy way to learn how to work with the system. And then Kubernetes self-integrates with the cloud that it runs on. So all of these things, obviously, storage has to be somewhere. So Kubernetes has a plug-in layer that will create the underlying systems for you. So we start Terraform to create the basic system, and then Kubernetes for everything on top of that. And in just everything is an API, whether it's application or infrastructure, everything is software defined, and it gives us a common language, okay? And so how it all ties together, I'm going to just summarize quickly because I think I'm taking up quite a bit of time. We have all of these different components, the different pods and volumes and things running. We had existing code to work from, but it was very poorly documented. Unfortunately, we had to do a lot of manual work to figure out how to make it all work together. So where do we want to be? We want to automate even further, okay? And we want to head towards what is called GitOps, which is that I said it's software-defined infrastructure, but now your software definition is in a Git repository. And if you want to make changes to your infrastructure, you change the code. And then the system redeploys based on whatever's in that repository. And Kubernetes is exceptionally good at this. You can knock out a piece of a Kubernetes, like you can knock out a worker node, and Kubernetes will then try and figure out, okay, well, I've lost these pods, let me go recreate them over there. So it's kind of quite self-healing when your cloud is healed, which is not happening with Elifu at the moment. It's the underlying cloud is a bit broken. And we want to implement all the automation logic in Kubernetes, so the scheduling of periodic jobs, or what you would call cron jobs and all of those kind of things, health checks, checking that the system is live, that the load is being handled correctly. And then we want to modify the existing system and add new microservices based on the additional capabilities that we want. So we got an existing system. If we want something new, we don't really want to change the existing system if we can avoid it. We want to just add another piece on the side that gives us the power that we want. So yeah, lessons learned and limitations. Working with other people's code can be both a blessing and a curse, even when you've got them on call. Decomposing these things requires developer commitment. We had some head-butting sessions with developers who were like, I just want to build one thing. I don't want to understand this whole mess of complicated things. And we had to explain how our needs as deployment team was actually ultimately going to make their needs as development team easier, not harder. And thinking about how to manage the infrastructure is paramount. Developers tend to just think about the application, but we brought the importance of, well, you actually need to manage your infrastructure in a way that actually gives you less pain over the long term. And in the future, monitoring, testing, and a multi-site deployment from GitOps. I said, remember, we want to have this running across multiple systems in multiple locations. So that is where we are heading. And yeah, so that brings me to the end of this talk. We have so many partners in this, here in the room, and many not in the room. But yeah, I hope that you will be inspired to go find out more about Kubernetes and this kind of DevOps way of doing things, and bring us into the 21st century of infrastructure deployment.
Thanks, Peter.
Okay.
Appreciate it. Any questions for Peter? Either roving mic. Or anybody else?
Stig has got a question.
Great talk. I might be bowling you a slow one here, Peter, but did you pay for any of this stuff? Or is it entirely free and open source software that you built this environment from?
We didn't even pay for Eugene's time. I shouldn't say that with the CHPC around me, but yeah. Yeah, no, this was 100% open source based. Look, for the production, we are considering our options, but for a development environment where we don't need to burn holes through credit cards, then yeah, we could go open source all the way.
Nice, yeah. I'd say if you didn't pay for his time, you probably owe him a beer then.
I probably do.
So, I know you mentioned one of it in your presentation, but my own concern is about documentation and sharing. I know we have that big problem as developers. But should somebody, a partner, want to replicate what you have done? Somebody has listened to you now and feels, oh, this is good. How do you share? How do you, we have this problem of documentation.
Yeah, so I see it in two layers. The one thing is really automate everything. So don't do what our partners in Canada did, which is kind of give you half of the system with lots of kind of holes in the, not only in the description of what various values meant, but also in the, you know, when we were trying to understand how to do something, occasionally they would say, oh, well, we have to like manually run this command. No, don't do it that way. So make it all automated, okay? But that is not documentation. And then yes, document as you're going along. We do have documentation in the sense of readmes at the moment, but also that said, I do believe that documentation to some extent is its own job. And we have the prevailing weakness of academia that you're always doing this work with a quarter of the workforce that you really need. So I'm not very proud of our current state of documentation. We need to do more of it. We have a technical specification documenting everything and how it all kind of relates together. We have better documentation than what we were given. But I agree that it's an ongoing challenge to make it live and up to date. And then, of course, you've got the problem that when you're changing infrastructure, the documentation needs to stay synchronized.
Okay, thanks. I think we need to move on, unfortunately, but thank you very much.
Now, everyone can hear me. Sorry for that. Okay, that's popped up there, but that's all right. Let me just make sure that I'm... All right, okay. Whew. Thanks, everyone. I am Patricia Swart from Diplomix. I'm the Bioinformatics Coordinator. And today, I just want to give an overview of what Diplomix is all about and what we do and how we work with the NICIS team. Let's grab this. It's not my day. I'm just going to stand here. No. Oh, there we go. And now it went. Too many slides. That's all right, I'll just stand here. All right, so Diplomix stands for Distributed Platform in Omics, and it's funded by the Department of Science and Innovation's South African Research Infrastructure Roadmap Program. And we have three main domains, those being genomics, proteomics, and metabolomics, and then bioinformatics. So we study we enable, rather, the studying of a whole bunch of biological data. So we have two sort of main objectives, the first being to build a world-class infrastructure, and the actions involved in this is to buy and strategically place instruments, help the service labs with their service and maintenance warranties, and provide advanced technical training. These have direct impacts, which is creating the availability of cutting-edge technology, building the confidence in the infrastructure system, which results in high-quality research. Over time, though, I don't know.
The problem is it's not, the down arrow not working. Just try this again. The problem is actually with the slide thing.
There we go. What did you do?
It's not working.
Okay. All right. So over time, we've focused on our second objective, which is now that there's this infrastructure in place, we need to enable access to this infrastructure. So that's increasing our training and outreach. We have spatial transformation programs, and then we have what we call high visibility, high impact projects. These result in increased skill development and increased awareness, and overall, we want to bring reputational gain to the South African omics community. Which button am I pressing?
No, there's a problem with forgetting that you are. The thing is that this mouse, it keeps opening another window on top of this thing. Is there another mouse attached to this thing somewhere? Because it keeps opening the. Can we disable track?
Can I unplug this?
No, it's not.
Because it's
Yes,
this.
yes. Let's unplug that. Yeah, it keeps. Oh, that's it. Sorry. Wait for that. Again.
Patience is key. I think we are working. Fantastic. Phew, presentations are fun to begin with, never mind technical problems. Okay, so that is that. But to achieve these goals, we don't do it alone. We're a small core team, so we have a network model. So on the genomic side, let me just start off by saying that we are hosted by the Center for Proteomic and Genomic Research in Cape Town, and CPGR. So CPGR is one of 11 network service labs in the country. So as I said, we help these labs by placing instruments, so sequencing instruments. We help them with their service and maintenance warranties, and also provide a little bit of wiggle room for them to do method development and optimize their systems and processes to ultimately lower the cost to genomic services in South Africa, so that we can keep genomics, the skills and expertise needed to generate genetic data in country instead of sending it overseas. As soon as you send something overseas, the skills and the people with those skills follow, and just builds that vicious cycle. So if you want to know what capabilities there are, if you want to conduct a genetic experiment, you can go to our website and you'll see all the capabilities. So we can do all of this in country, which is really fantastic. So you don't really need to send your samples overseas. So you can visit the website. Otherwise, you can get in contact with Dr. Shane Murray. She is the genomics coordinator at Diplomics, and she will be able to point you in the right direction. And then likewise, on the proteomics side, we have 3 core labs. Decipher is one of them, and that's a merger of CPGR and UCT's IDM. So they've come together to form Decipher. Then we have services at West University and at CSIR. And the contact person there is Dr. Liam Bell, and his team can help you with your proteomics and metabolomics experiments. So now you have these machines that are generating genetic data and proteomic data, and you need to figure out, okay, cool, what's hidden in this biological data, what's it's telling us, what's the story? And so that's where the bioinformaticians come in. In Diplomics, this is the Diplomics bioinformatics team. So it has myself, Sichaba, who is here, over there, second row. And she is an active practicing bioinformatician. Whereas I said I'm more on the coordination side. And then Mlakazi, who's also a bioinformatician, but she also has, part of her job role is to promote training specifically to bioinformatics. So as well, the bioinformatics branch of Diplomics is not structured in the same way as the genomics and proteomics side, because there isn't really a service or network rather of bioinformatics service labs or services in country. Again, many reasons. The bioinformatics as a job role is a bit of a gray area in some spaces. People aren't willing to pay for their bioinformatics services. Sorry, I feel like I'm shouting. So people don't want to pay for the bioinformatics services. It's often an afterthought when designing an experiment. And the postdoc can just do it. So again, yeah. So again, people with the skills get taken up by other industries again go overseas, cycle continues. So a few years ago, we envisioned and initiated Clarity. So this is a bioinformatics marketplace that we've made possible through some funding from Diplomics, well, DSI. And it's a space to promote bioinformaticians, to highlight their value, to pay them for their work. So our consultants are part-time. They're often postgrad students, PhDs, masters, postdocs, and early career researchers, who are just making their way through a very tough landscape. And then if you're on the other side, if you're a client, if you, like many, you end up with a huge data set in your lap, and no one's really thought about, okay, what we're going to do with it now. So preferably that's not what you do. Best case scenario is that you get hold of myself, either on either of those two email addresses, and we set up a consultation so you can discuss your omics project at the planning phase, so the design phase of your project. So get a bioinformatician involved from the beginning. This is a free 30-minute conversation. But either way, we will do our best. If you already have a data set, we can still help you out. So it's a sort of simple process, conversations with expert bioinformaticians. We can provide a full analytical report and results, but that's not really good enough in some ways, because we don't just want to be a black box. You don't want to just send data somewhere and then get a report. So a new thing that we've offered, and it's literally only come in the last month, because we've have more capacity now. We have more bioinformaticians on the consultant side. So we want to allow for one-on-one contact sessions. So we can do the analysis, but then we can work with you over a period of how many weeks to equip skills and and provide a better understanding. Yeah, so that is Clarity. So get involved if you want assistance, or if you are a bioinformatician and you want to build up a portfolio of projects, get more experience, or even just simply need a little bit more extra money. So now, that's sort of just over broad lines how we analyze the data. We need people to analyze the data, and that's a very specific set of skills. In parallel to that, what do we do with the data? We need to move it. We need to move it from the sequencing device to where it's going to be analyzed, to where it's going to be stored. And yeah, so this is where we've been working with NICIS, and they've been incredibly helpful. But there is room for improvement in the relationship between the omics community and the NICIS services. So what we've done is over the last couple of months as well, we've initiated what we've called bioinformatics community group meetings, where people in the Diplomics network, not necessarily bioinformaticians, but even just people working with the data, to come together and discuss the challenges. And the first meeting we had, can't remember when exactly it was, but all these problems came up, the standard problems of how do we move this big file, haven't even thought about where we're going to store it long term. So what I did was we got some representatives from Sanren and Derisa to literally just come have a brief Q&A. They gave a brief presentation on the services, and then it was an open discussion about explaining the needs and what services, and just meeting halfway. Don't worry, Werner, we'll get to CHPC hopefully early in the new year. So NICIS and these services are crucial to what we do, and very important to the omics community. What I've discussed at the moment is sort of individual researchers. So a researcher goes to a service lab, they get their data, then they manage it in their own way. But let's think bigger. So if you remember way back with all those technical difficulties at the beginning of this presentation, I mentioned high visibility, high impact projects. I'm going to briefly touch on one of these projects, and this is One KSA. We are intending to sequence the genomes of over 1,000 South African species. It's called One KSA because it's a catchy name, but obviously there are more than 1,000 species in South Africa. And this is a subset of the African Biogenome Project, which in turn is a subset of the Earth Biogenome Project. There are obviously some key differences. We're focusing on South African species, and we have also chosen to go with Oxford nanopore sequencing technology. So currently it's in 2 phases. The first phase, which is running from September 2023 to March 2024. It's our method development and training phase. Very strong, heavily weighted to those departments. We're sort of learning on the go, and yes, I should mention, again, this is a very ambitious project. It's not going to be done in one lab. It's going to take a network. So we're going to use our network. And so we've brought in PromethION sequencing devices. We have a P24 in Cape Town at the CPGR. We have one at P2 Solo at UWC, UPGL in Pretoria, and then SciAb in the Eastern Cape. So those are our service labs. So researchers can send their samples to those labs and get their species sequenced. Phase 2 will build on the foundations of phase 1. We really need to build some solid foundations there. And it will have a citizen science component as well as an open science component. So open science, I just want to mention, it's not going to be freely downloadable. That's not necessarily what open science is. We want to make it known that these data have been generated, and then after that there could be a data access committee where you can motivate to use those data. That's the plan there. So phase 1, also how and what do we do with all these data that these sequencing machines are generating? As we have at the moment 4 labs generating data. This is broad strokes. The data can't stay on the machines, so it needs to go to some in-house storage to begin with. Then it's going to go to the CHPC for genome assembly, and then also concurrently for short-term storage at Teresa, and then long-term storage at Teresa as well. And then the way we share the data back with the contributor, because the data will belong to the contributor, that will also be through Teresa. So this is a biodiversity project, but the challenges in terms of data transfer and storage and the compute power that we need to assemble these genomes is the same across other projects. So what we learned from this, we can apply to a whole bunch of other projects and keep it in country. I think that's key. That's key for me, is that especially for the biodiversity project, it's South Africa's biodiversity. We want to create a national asset that can be used by South African scientists for South Africa. So that's the sort of core heart of the project. But then, yeah, like I said, what we learn from here, we can apply to other projects. Like, there's a 100,000 genome project on the horizon, and I think we're talking petabytes of data there. It's going to be a challenge to do that, but I believe that we're up for it. And yeah, just huge thank you to everyone at NICUS who've been very patient with some of my questions. And yeah, really look forward to working with them further, and yeah, just strengthening the relationship between NICUS and the omics community in South Africa. There's a nifty code for you if you want more information to links. And where did that go? Cool. Thank you very much. Thanks for listening.
Thank you very much, Patricia. Yeah, fortunate for the delays in this topic.
That's okay.
It's really important work, and I think Nikki's and certainly CHPC is proud to be associated with this, and we will build this partnership. CHPC has been in MOU with Diplomics for some time, but it's basically on paper. It's something that
Yes.
needs to materialize much more, and obviously this is going to happen in the initiatives that you're talking about. But the floor is open. Any questions? Either one. There might be. We're just going to refer to the mic people needed, but I thought it sounds quite well. So I know you're the bioinformatics manager, but in terms of the omics service providers, I had a look at the websites, and it's all links to the existing websites. How do we know, as somebody who doesn't have, because my understanding is that one of the reasons the diplomics exists is that we don't all have to buy our own sequences, right? But how do we know how much is there a price list for getting certain things sequenced, or how does that model work?
So you would have to contact the individual labs for that price.
And on that, because some of the labs, unfortunately, I've heard very bad stories about the abuse of other people's samples when they get their hands on it. Okay. So, do you have a standard? I mean, does Diplomics have any standard agreements on the expectation of that samples will only be used for sequencing and not kept, and all of those kind of things? Do you have those kind of agreements with your labs?
So the individual labs will have a sort of service agreement, and they should be very clear on what happens to those samples. I can really only speak for CPGR. No, I can't actually speak for CPGR, but I know when you send a sample to CPGR, you say what must happen to those samples, and they do honor it. In terms of one KSA, we will have a sort of sample submission form, which will also detail what will happen to the sample, what we will use it for. And it is very clear, and it is a legally binding agreement.
Thanks.
Last question from.
So it's, hello? Hi,
Yeah.
I think we know each other.
Yes, we have made.
So I am on the ethics committee of the Earth Biogenome Project. And it is where it's being tried to sequence pretty much all the genomes of all the organisms, which is far-fetched, but maybe in the future, where the discussion is going on. Where are the samples are coming from? What is being, and what documents need to be signed in order that it is made clear that benefit sharing, for example, is part of the agreement. Where the documents of sampling, whether you have a permit or not, is being clear. Where traditional knowledge holders are clarified. And even in that environment, it's not straightforward. Let me just mention that the majority of the council members are lawyers from the United States. So I know that I know that this sequencing of the plants, of the native flora, should be done in South Africa. I am sure that the data needs to be secure, that access must be verified so that benefit sharing agreements are signed. But this also implies that before this is done in large, it must be absolutely clear who is sending you the data. These rules, before you start opening any bioinformatics or opening any services, this must be clarified. You must have an ethics committee, if possible, with the government of South Africa, because if you look at rooibos, so you know that I am sequencing the rooibos genome, that there are reasons why the legislator has been established for rooibos to be, that there is a benefit sharing agreement between the rooibos industry and the knowledge holders. So I think before you offer, you must establish an ethical background for all the documents, and that must be, you must have a legal team on that. This is not something which you can play around with, because except for rooibos, none of the other medicinal plants are protected, or not even medicinal, none of the other endemic plants are protected from being
exploited. Yeah.
So I would love to discuss this with Diplomix further.
And we would love to discuss it with you as well. Yes.
Thank you very much.
Thank you, Liana.
Unfortunately, the time has gone out, but thank you very much, Patricia. We appreciate that.